Welcome to this section on molecules in motion. We will begin by discussing the small signaling protein ubiquitin. Ubiquitin is very unique in that it can form polymeric chains on itself at 8 different linkage sites, which then can be attached to substrate proteins for various signaling outcomes. If you can imagine one ubiquitin as Medusa's head in this picture, 8 other ubiquitins can also be attached to it at the same time. This is accomplished using a special chemistry which involves the C-terminal tail of ubiquitin being covalently linked to lysine residues on either another ubiquitin or a substrate protein. If we take a look at how these linkage sites are distributed on the surface of ubiquitin, the 8 are methionine 1, lysine 6, lysine 11, and on the alpha helix, lysine 27, lysine 29, lysine 33, lysine 48, and on this loop region we have lysine 63. There's also a very important hydrophobic patch made up from 3 different hydrophobic residues. Depending on how a ubiquitin chain is formed, many different signaling outcomes can occur. This happens to depend on the linkage that is in the chain. As we can see on this slide, the outcome from lysine 48 linkages is very different from those of lysine 63 linkages. So how is this even possible? Here at the University of Maryland, we have used nuclear magnetic resonance to uncover how different ubiquitin linkages can result in different signaling pathways. The work is pioneered by Professor David Fuchsman, who has found very distinct structural properties of lysine 48 and lysine 63 linked ubiquitin chains. Let's have a closer look at lysine 48 polyubiquitin chains. There are many possible conformations ubiquitins can sample when they're linked to each other. However, for lysine 48 linkages, it has been narrowed down to this structure, which has the hydrophobic patch residues forming a very tight interaction, and this causes the chain to adopt a compact shape. However, the receptors also need to recognize these same residues that are buried by the 2 ubiquitins. And there are inherent dynamic properties which allow the ubiquitins to open and close. When the lysine 48 ubiquitin chain opens and closes, receptors, such as the receptor shown here in magenta, can interact with the hydrophobic patch. This allows for proper signaling to occur. In stark contrast to the lysine 48 linkage, the lysine 63 linkage, using a different site in ubiquitin, results in a completely different structure for the chain. As we can see, the hydrophobic patch residues are nowhere close to contacting each other, and the chain is very extended compared to the lysine 48 linkage. The lysine 63 linkage also has its own inherent dynamic properties. However, the structures it populates never form the compact conformation seen with the lysine 48 linkage, and the chain always remains extended. Receptors that interact with lysine 63 linked polyubiquitin chains do so by recognizing the extended structure of these chains. This is in contrast to receptors for lysine 48 linked polyubiquitin chains. Based on the structural properties of these different linkages, this explains how one protein, ubiquitin, can take on so many different functions in the cell.